Alright guys, the previous video I mentioned about how non-specific the rubisco is. It make a lot of error rate. So in some plants that live in some area that get more errors of rubisco, they evolve to have a workaround mechanism to make the error rates go down. And those kind of carbon fixation is called C4 carbon fixation and CAM photosynthesis, for example. Here you have C3 plants that use only Calvin-Benson cycle to fix the carbon. For C4 and C4 and CAM plants, these two mechanisms fix carbon twice. The first product is carbon-4 compounds. So this is called C4 plants. It fixes the carbon dioxide in the first time into C4 from C3. And this C4 can travel from one place to another. So the two fixation occur in different cells, different space. This C4 travel into the inner compartment of the plants, and then it releases carbon dioxide for Calvin-Benson cycle. And the C3 carbon can be recycled back to the first compartment. For CAM plants, the similar things happens, except the first fixation and the second fixation didn't happen at the same time, but everything's occur in the same space. So C3 at night, it will fix the carbon dioxide to become C4, and then this C4 will be stored in vacuole. During the daytime, this C4 compound can release carbon dioxide for Calvin-Benson cycle fixation, and C3 is then stored in the form of starch. And this starch during the night, it can release C3 compound and fix another round of carbon dioxide to become C4 again. So let's get into the detail of C4 photosynthesis. So C4 photosynthesis is evolved for the plants that live in an area that has higher temperature, because higher temperature lead to more errors of rubisco. And it is also help the plants that live in where the carbon dioxide concentration is lower. So most of the crops, such as corn, sugarcane, sorghum, use this C4 photosynthesis as a mechanism to enhance the capacity of rubisco. The primary products of carbon fixation is C4 compound, and in this case is oxaloacetic acid, or we can shorten it as OAA. C4 cycle is mediated in different compartments, and those compartments have structure right here. If you cut the leaves of C3 plants, you will see some structure like this. You have parasite mesophyll, you will have vein with xylem and phloem, you have bundle sheet cell around the xylem and phloem, and also you have spongy mesophyll scattering around. And notice you have stroma here. Stroma is like a window that allows oxygen to get out and carbon dioxide to get in for photosynthesis. So let's take a look at the C4 plants. Here you have epidermis, here, and you have stroma. Instead of having parasite mesophyll and spongy mesophyll. You have the mesophyll that wrap around the bundle sheet cells. And this structure, we call it Kranz anatomy. Kranz in German is just mean reed or like a ring around the bundle sheet cells. So the first carbon fixation process is happens in the mesophyll cells where you start with C3 carbon, C3 compound, 3 carbon compound with carbon dioxide, and then you get the oxaloacetic acid as the products, and then oxaloacetic acid move to the bundle sheet cells and release carbon dioxide for Rubisco to fix the carbon for the second time. So you can see it more clearly in this picture. Carbon dioxide in the air get fixed, but actually it will become a bicarbonate. You can see that it has only one carbon, and this bicarbonate can be fixed by phosphoenol pyruvate, which is the carbon 3 compound, 3 carbon compound, and it will become oxaloacetic acid or C3 compound. This is the first carbon fixation or primary carbon fixation. This C3 compound from the mesophyll cells outside, closer to the stroma, it will travel through the plasmodesmata between cells, and it will release carbon dioxide for Rubisco to fix it for the second time. The C3 products will travel back to the mesophyll cells, where it can be regenerated and fix more carbon for bicarbonate again. All right. Here is the efficiency of C3 and C4 carbon. So we have two situations. The left panel is the light intensity and the increase in photosynthesis. The right panel, we are considering about increase in temperature and again, the photosynthetic rate. You have two lines of graph, C3 in the pink and C4 in violet or purple. So when the higher light intensity, C3 plants stop making more photosynthesis very early, but C4 plant can still making more photosynthesis until at some point they stop as well. This means C4 has more efficiency for this. But in the lower light intensity, you can see that C3 is actually better. Similar to higher temperature, C3 has lower capacity in the very high temperature above 25 degrees Celsius has lower capacity for photosynthesis, but C4 is very consistent. You can see that at lower temperature, at lower light intensity, C3 is actually do better than C4. This is because during the C4, you need more energy to convert the C4 to C3 and for C3 to recycle back for this process again. So you need more energy for that. So if there are not too much of the burden, the plant should stay in the C3 region, but it will help in some area that very hot and very bright, something like California or Provence in France, that there is not much of the rain, not much of humidity, and also a lot of sun, high temperature, for example. All right, the last one, which is CAM photosynthesis. This is called CAM because it actually discovered in the plants family that's called Crustulaceae. I'm not sure if I pronounce it correctly. So the full name of CAM photosynthesis is crustulation acid metabolism. Mostly the plants in this family are succulents like cactus, or this one is agave or agave, and pineapple. These plants mostly stay in the very dry area. It needs just very little of water, mostly in desert where um, plants need to conserve energy to conserve moisture or water from drying out because they don't have much of the water available. So plants in this area tend to close their stoma or stomata during the day in order to reduce water loss, because during the day you have higher temperature and the plants will lose more or, of moisture or humidity from transpiration, sorry. So the plants close the stomata during the night, but plants close stomata during the day to conserve water, and during the night they open their stomata in order to exchange the gas. This is in opposite of the C4 and C3 plants that open the stomata during the day for photosynthesis and close the stomata during the night because there is no light for photosynthesis. But for CAM plants, they open their stomata during the night and close it during the day to conserve water. So in order to make photosynthesis happens, because as I mentioned back to some video ago, that fixing carbon need light, because light you will get NADPH, you will get ATP, and most of the enzymes in the carbon reaction need light to activate them. But for the first fixation of carbon in the CAM plants, those enzymes do not need light to activate them, so they can do it during the night. So what happens is carbon dioxide from atmosphere during the night get into the stomata and got changed into bicarbonate, and also during the night mitochondria produce more carbon dioxide as well. This bicarbonate is then fixed with phosphoenol pyruvate. Together, this is a C3 compound and become oxaloacetate. And this oxaloacetate can be reduced to form malate. The reduction of oxaloacetate need NADH. This malate can be stored as the malic acid in vacuole. And this malic acid during the day, it will be released from vacuole and become malate. And this malate can be oxidized. It gives an electron to NADP plus and release carbon dioxide for Calvin-Benson cycle that will fix the carbon during the day to become 3-phospho to become glyceraldehyde 3-phosphate, and that glyceraldehyde 3-phosphate can be used to make 6-carbon glucose, and that glucose can be stored in the form of starch right here. During the night, this starch is degraded and you will get triose phosphate or 3-carbon sugar, and that will be recycled to be phosphoenol pyruvate that can be used for fixing more of the bicarbonate again. So this is all about photosynthesis. And so I hope you guys are still with me. And if you guys have any question or if you don't understand anything, please feel free to email me. All right. And I hope to see you on Monday.